Hello, my name is Mounika Boddireddy. I work as a bioinformatician in Lonza Biologics R&D, Cambridge, UK. My poster title is Enhanced Antibody Production through Multi-Omics Analysis Leveraging Long-Read Sequencing Technology. First, I would like to start with a brief background about the poster and the analysis I have done. Transposon-mediated transfection is one of the efficient approach for the biologics production in host cell genomes. So these transfection methods are used for inserting the foreign DNA in host cell genomes. However, the location and number of transposon copies in the host cell genomes is not known clearly with the methods which have been implemented so far. So the main aim of my poster is to accurately quantify these transgene insertions and that to full-length transgene insertions in the host cell genome. For this, I used a nanopore ultra-long read sequencing data and also integrated these nanopore data with multi-omics analysis of RNA sequencing and ATAC-seq data in order to identify the expression patterns of the transgene insertions and their correlation with the host cell gene expressions. So in the method section, I would like to briefly explain about the bioinformatic analysis which I have done. So in the bioinformatic analysis, I performed mappings of the long read sequencing data against the transposon vector sequence and also the reference host cell genome sequence, which is used for the quantification of genome-wide insertions. And after identifying these genome-wide insertions, we correlated with the insertion sites with the RNA sequencing data and also ATAC sequencing data. Now I would like to explain the results and the interesting observations which I have obtained in my analysis. So long read sequencing confirms the full-length transgene insertions without any vector backbone insertions where the highest levels of mRNA is observed for the transgene. So in the plot here, we see the first plot is the whole genome sequence for the ultra-long nanopore read sequencing data with the coverage of the number of reads which is mapped on the y-axis, and on the x-axis it's the vector scheme which we can see here below the plot. So the transgene insertion is flanked by the ITRs, and there is also a vector backbone here. And this vector schema corresponds to the positional coordinates on the x-axis. And the second plot is the RNA sequencing coverage, which is mapped for the same vector map along the vector map positional sequence. So we can clearly see that in the ultra-long nanopore whole genome sequencing data, there is high coverage for the vector map between the region flanked by the ITRs, and there is also correspondingly higher mRNA coverage in the RNA sequencing data in the region of this ITRs, which is the transgene. And in the analysis of transgene insertion sites integrated with the ATAC-seq sequencing data, we can clearly see that all the peaks which are highlighted in red here are the transgene insertion sites, and all the black peaks are the ATAC-seq sequencing data. And we can see that these insertion sites are preferentially occurring in the open chromatin regions with respect to the ATAC-seq data. And all the peaks which we see here are surrounded by these coverage peaks from the ATAC-seq data in most of the insertion sites, which are the transgene insertion sites in the host cell genome, and these are the scaffolds of the host cell genome. And on the x-axis, it's the positional of the scaffold, which is plotted in a million base pairs. And in addition to this, we also performed the expression the influence of transgene expression on the nearby genes. So we extracted the upstream and downstream genes to the insertion sites in the host cell genome, and we plotted the TPM of the expressed genes. And we clearly see that there is no significant effect of the transgene insertion sites on the genes which are located both upstream and downstream when compared between the transfected and non-transfected clones. So the transfected clones is in red color and the non-transfected clone is in blue color, which we can see here. So it's the upstream and downstream genes which are located with respect to the transgene insertion sites. So after obtaining all these results, I would like to briefly conclude about the key findings which I have obtained in my analysis. So using long read nanopore sequencing data, we managed to successfully identify the full length insertion sites inside the host cell genome, and we also got this interesting observation about the ATAC-seq data where we identified the insertion sites in the open chromatin regions. And as said earlier, there is also no effect of these transgene insertion sites on the expression of the genes which are located nearby. So the final takeaway message from our analysis is that exploring the impacts of transgene insertions by taking advantage of ultra-long nanopore read sequencing and integrating it with multi-omics analysis can give us more in-depth insights into the expression profiles of the transgene insertion sites inside the host cell genome and where they preferentially occur, and which can be used for the effective production of biologics products by making use of this information. Thank you.